Assalamualaikum and hello everyone. Today, me and my friends will explain about the diversity of fungi. Did you know guys what is fungi? Basically, fungi are eukaryotic organisms that include microorganisms such as yeast, mold, and mushroom. Now, fungi are considered to be the closest multicellular relative of animals. Did you guys ever seen fungi in your daily life? So here, I will show you one of the fungi that we can see in our daily life. It is bread mold. There are many types of mold that can grow in the bread, such as aspergillus, mucor, and rhizopus. There are also many types of fungi that grow around us. So, stay tuned to know more about the diversity of fungi.
Hello everyone, my name is Noashan binti Muhammad Skifli. So today, I will explain to you about the fungi with the zoospores. There are 3 phyla of fungi with zoospores, which are Blastocladiomycota, Neocalimysticomycota, and Chytridiomycota. So let's get into the Blastocladiomycota. There are many examples of the Blastocladiomycota, but you can see some of the examples here. So let's get to know about the characteristics of this fungi. Blastocladiomycota is actually lives in the freshwater habitats, but you can also find them in soils and muds. They act as the saprotrophs, decomposing plants and animal debris, and also parasitize the arthropods. Some of the fungi are also pathogens of the small aquatic plants and animals. So how do they produce the new substance? As I said before, that these fungi will produce zoospores. They will undergo sporomyces and form an asexual reproduction, which are known as anisogamy. This reproduction consists of the fusion of 2 gametes. Many of the Blastocladiomycota decompose cellulose and other dead organic matters, but most species are parasites of plants, invertebrates, and algae. Here are one of the examples of Blastocladiomycota, which is Physoderma mydes. It is a serious plant pathogen that causes the brown spot on the leaves of the corn. Do you want to know that? In the previously, Blastocladiomycota are considered as the same group of Chytridiomycota. The only main difference between Blastocladiomycota and Chytridiomycota is that the Blastocladiomycota can produce an extensive mycelium. So next, let's get to know more about the other type of fungi, which is Neocalimysticomycota. Here are some of the examples of the Neocalimysticomycota. So for the characteristic, I think that there are some similarities with the Blastocladiomycota, which is Neocalimysticomycota can also produce mycelium, but it tends to be smaller than Blastocladiomycota. It can also produce multiflagellated zoospores. One characteristic that are unique about this fungi is that it lacks with mitochondria, but it has the presence of the hydrogenosomes. The presence of hydrogenosome can actually reduce the mitochondria that have adapted to their anaerobic environment. So, this Neocalimysticomycota lives in the ruminants and other herbivores. So, they will break down the cellulose and other indigestive matters. The genome of the genus Neocalimysticomycota was found to have genes that code for digestive enzyme that only present in the bacteria. The genes were obtained through a process of horizontal gene transfer by sharing DNA between unrelated species. Due to their ability to degrade cellulose at high temperature, they have the potential to be used in the biotechnology for production of biofuel.
Now, I will explain about the Chytridiomycota. The chytrid, which mostly aquatic fungi with regulated spores that may represent the most ancestral fungal lineage, include the simplest fungi, which may resemble the first fungi to have evolved. Some chytrids are single cells, others from branched estate hyphae. Most chytrids reproduce asexually through the production of zoospores within a single cell. Then, the zoospores grow into new chytrids. Chytrids also play a role in the decay and digestion of dead aquatic organisms, but some are parasites of living plants, animals, and protists. Did you know guys, the parasitic chytrid, which is Batrachocytrium dinodrobatis, in the last 30 years has decimated populations of over 200 species in frogs, resulting in a staggering loss of amphibian biodiversity. The chytrid infects the frog skins, causing a normally permeable layer to thicken and inhibit the oxygen and water intake. The electrolyte imbalance results in cardiac arrest and death. So here, I will explain about the Glomeromycota. The Glomeromycota or AM fungi are a relatively small group which is 230 species of fungi. The name of AM stands for arbuscular mycorrhizal fungi. AM fungi are branching invaginations that the fungus makes when it invades the plant roots. Mycorrhizae are mutualistic association of plants and fungi, and AM fungi are the most common fungi to form symbiotic relationship with the plants. They play a critical role in the ability of plants to absorb nutrients with their roots. The majority of plants have a mutually beneficial relationship or symbiosis with the egg and fungi.
Well, the next fungal lineage is Zygomycota. What is Zygomycota? Zygomycota, usually known as the black bread mold or its scientific name called Rhizopus stolonifer. We usually could see this fungi on rotten vegetables, fruit, and bread, like this. Now, let's move on to the characteristic and the structures of Zygomycota. Firstly, most of the Zygomycota are saproots, which means that they can decay organic material. Secondly, they can produce both asexual and sexual according to the environment state. Asexual reproduction occurs when the environment state is favorable, while the sexual reproduction occurs when the environment is not favorable. Thirdly, some of the Zygomycota are infectious to animal and human that can cause disease such as gastrointestinal. Fourthly, they have a quinocytic hyphae, not septated hyphae, with haploid nuclei. For the structure, we have sporangium, spores, sporangiophore, hyphae, and rhizoids, which hyphae that grows into bread and carry out digestion and absorb nutrients. Next, we move on to the life cycle of Zygomycota, which contains asexual and sexual reproduction. In asexual reproduction, spores are produced from a haploid sporangium by mitosis. Then the spores will disperse into air and land at a suitable place such as bread to germinate new hyphae. Then the new hyphae will produce more sporangium and more spores. Sexual reproduction starts when two opposing mating strains plus and minus mate, and they produce isolated capsule called gametangia. Then the plasmogamy occurs as the gametangia fuse each other to form a zygote. Then the karyogamy will take place to form a diploid zygote. The cell walls of the diploid zygotes are thin at first, but later thicken into zygospore. And the germination begins when the diploid nucleus undergoes meiosis and forms sporangium at the end of the germ tube. Then the spores will produce within the sporangium. Now, I will explain to you about Ascomycota, the members of the phylum usually called as the step fungi. Here are some examples of the Ascomycota, which are Marcella, Peziza, and Saccharomyces, which is yeast. Now, let's move on to the characteristics and structures. Firstly, Ascomycota can produce both asexually and sexually. The sexual reproduction occurs in the fruiting body or ascocarps, like the picture here. Unlike Zygomycota, Ascomycota have septated hyphae. And lastly, they are essentially important for recycling, production of food such as bread and cheese, and production in making antibiotics. Now, we move on to the Ascomycota life cycle. In asexual reproduction, spores called conidia spores are dispersed into wing. Conidia spores are produced from the haploid conidia. Conidia can develop at the tip of the conidia spore. Sexual reproduction occurs when two septated hyphae from different mating spores meet. Then, plasmogamy occurs as the two hyphae fuse together, forming a dikaryotic cell. They produce hyphae containing two haploid nuclei in one cell. Then, karyogamy occurs as the two haploid nuclei fuse together, forming a diploid zygote. Then the diploid zygote undergoes meiosis and mitosis, forming 8 haploid ascospores within the ascus. Lastly, ascus will mature, then release the ascospores into the...
Hi everyone, my name is Nurhana Humaira binti Azman, and today I will explain to all of you about fungi in food industry. When you heard of bread, what comes to your mind first? Is it soft, fluffy slices, or delicious? After you mix all the ingredients together, you have to wait for a while for the dough to rise before you can bake it into a yummy bread. Why do you have to do so? What's going on that caused the bread dough to puff up and rise? That is right. It is because of yeast or Saccharomyces cerevisiae sugar-eating fungus. In bread dough, the yeast cell mix with warm water and begin to feed on sugar. As yeast cells feed on sugar, they produce carbon dioxide gas and ethyl alcohol in chemical process known as fermentation. The carbon dioxide gas released by the process of fermentation get trapped in the sticky elastic dough, cause it to puff up or rise. This can take a while, and that is why you need patience when you are baking bread. Likewise, the ethyl alcohol produced give the bread its unique smell and taste. Before you say ew, you need to remember that fungi can be very helpful and as well as tasty. You need to know that mushroom on your pizza are fungi too. It is not just that. A mold called Aspergillus is used to produce soy sauce and Japanese food miso by fermentation of soybean. Not to forget,
Your favorite cheese such as blue cheese, Roquefort, Camembert and Stilton cheese are ripened with the help of mold called penicillium. But you need to remember, mold can also be detrimental to food industry. Peanut and other grains are susceptible to colonization by certain species of aspergillus that produce aflatoxin, which can damage an animal's liver. That is why the US FDA has established guidelines on the amount of aflatoxin allowed in food consumed by human and livestock. Now, let's move to the next topic on fungi importance on human. Fungi can be beneficial to human but at the same time also dangerous. Fungi interaction can transform from commensalism to parasitism depend on host, environmental and fungal factor. Candida albicans is a yeast that is normally found on the body. But how can fungi be harmful for humans? This happens when the balance of Candida and other microorganisms is disturbed. The yeast proliferates, causing oral thrush in newborn and vaginal infection in women. Have you ever heard about tinnitus infection? Actually, tinnitus is an infection of the skin that caused by the fungi. The symptom of tinnitus infection is feeling itching, peeling skin, or a rise inflammation. Athlete foot, dog itch, and ringworm are the example of tinnitus infection. Did you know that it is difficult to design fungal medication that do not harm patients due to the strong similarities between fungal and human cell? One positive medical benefit is that many said fungi have antibiotic and antimicrobial properties. Certain members of the genus Penicillium can be used to produce the antibiotic penicillin, which is a treatment for bacterial infection. Another produces cyclosporine, which control the immune system to prevent organ rejection after a transplant. Many geneticists also use Ascomycetes as model organism. With genomic information available, Aspergillus nidulans serve as a safe model for more dangerous related species. That is why Neurospora crassa was used to develop the one gene, one enzyme hypothesis. And Saccharomyces is a single-celled eukaryote with genes similar to those other eukaryotic organisms, and they can be easily manipulated in a lab.
So, hi guys, my name is Dini Haikal. So today I'm going to explain about Basidium mycota. Basidium mycota consists of 31,000 species. For example, mushrooms, toadstools, and puffballs. Fungi that cause plant disease also placed in this class. But not all fungi are poisonous. For example, such as Portabella and Chaete are served as food by humans. There are 75 species of Basidiomycetes, which is poisonous. So we go to the first example, which is Amanitas. Amanitas species have two colors, which is yellow and red. This species also called as fly agaric because it kills flies. It contains a toxin called muscarine and muscaridine, which produce symptoms of alcoholic intoxication. In 1 to 6 hours, victims will lose consciousness and suffering hallucinations. Luckily, it also causes vomiting, which makes the death occur from this species is less than 1%. So the next one is Amanitas phalloides. Amanitas phalloides cause 90% of fatalities to mushrooms poisoning. It contains a poison which is called amanitins, which will inhibit RNA polymerase. This will cause victim to die due to liver and kidney damage. So the next one we will go to Psilocybe mexicana. Psilocybe mexicana contain psilocybin, which produce dreamlike state, which a vision of colorful patterns and object seems to fill up space. Not just that, this species also sharpen other sense ends and the victim will feel intense reality. So next, we go to reproduction of Basidium mycota. Basidium mycota have basidium structure which spores called basidiospores develop. Next, they also have basidia, which are located within a fruiting body called a basidiocarp. Under the carp of Basidium mycota, it have gills. So the first step is basidiums produce mushrooms, or we call it as basidiocarp. Next, basidia will form a gills of mushrooms. In the gills of mushrooms, nuclear fusion happens in the diploid nucleus. After that, meiosis will occur and basidiospores are produced. After the basidiospores are produced, the basidiospores will release, which will make the germinations of spores results in haploid hyphae. After that, the cytoplasmic fusions resulting in the